So now that we have a good understanding of the four major steps associated with initiation, let's take a more intense look at the promoter recognition part of initiation. So, to orient you to this figure here, we've got the promoter region, which is going to be in yellow, and we have our non-template strand and our template strand of DNA. Our non-template strand is going to be oriented 5' to 3'. Our template strand is going to be oriented 3' to 5'. The promoter region is going to have two major landmarks associated with it in bacterial species. So this is going to be at -10, so 10 base pairs before the start site, and then another one at -35 or 35 base pairs before the start site. And then after the start site is going to be our coding region. So promoters have these consensus sequences, and these are going to commonly be encountered as nucleotides that are going to be at a very specific location upstream of the transcription start site. So the TATA box is the consensus sequence that we see at -10. So it starts out with TATA and then AT. So we call that the TATA box. And then the second consensus sequence that we see a little further upstream from the start site is this TTGACA promoter recognition. And those are going to be key components that RNA polymerase is going to be looking for as it sets down and associates with the promoter region. All right, so again, that bacterial promoter consensus sequence is located at -10, that's our TATA box, and then at -35, which is the TTGACA sequence. So I don't expect you to know the specific sequence at -35, but I do expect you to know that there is a consensus sequence at -35, and the TATA box shows up at -10. So one key aspect um, of gene recognition and control really comes down to some of the research that's been done with promoter regions. So if you're trying to suppress, expression of a gene, one thing that you can do is go into the promoter region and substitute nucleotides in these areas. And the transcription rates really drop off and it becomes very, very inefficient. So this is what these consensus sequence are what RNA polymerase is looking for. So it's looking for these two recognition sites so that it can know exactly when to start bringing in new nucleotides to form the messenger RNA strand. All right, so let's look at what step one or initiation looks like. So here in this figure, you're just going to have to stick with it for just a little bit. We're going to be moving from the top down. So up here at the top, we've got our double-stranded DNA, and at a midpoint in that DNA is going to be our promoter region, and at the very end of it is going to be our transcription start site. So floating around in the nucleus is our core RNA polymerase, and so that core RNA polymerase is going to bind with the sigma factor. And once that sigma factor associates with the core enzyme, it's going to form that holoenzyme. So that holoenzyme is when the sigma factor is actually attached to the RNA polymerase, and it's going to, that whole unit is then going to bind to the -35 and -10 consensus sequences in the promoter region that we just talked about. After that has happened, then the core enzyme, the holoenzyme, is going to bind that promoter region very tightly and begin to unwind the double-stranded DNA, creating that replication or that transcription bubble or that open complex. Then if we look over here at step number 4, that nucleoside triphosphate is that is complementary to the DNA at the start site is going to serve as the first nucleotide in the RNA molecule. So the two phosphate groups are then going to be brought in. They're eventually going to be cleaved from the subsequent RNTPs, creating an RNA nucleotide that is added to the 3' end of the growing RNA molecule. So we can see that actually starting to grow here. And finally, the sixth step is that once that mRNA or that RNA polymerase gets started with the creation of the mRNA strand, the sigma factor is going to be released, and the RNA polymerase is going to move beyond the promoter region to create that messenger RNA. So the take-home message from this initiation step is that RNA transcription is going to be initiated when the core RNA polymerase binds to the promoter region with the help of the sigma factor. That leads us into step number 2, which is elongation. And here it's going to be pretty straightforward as it was in DNA replication. So elongation and transcription is where RNA polymerase is going to move downstream. It's going to continue to unwind the double-stranded DNA at the leading edge of that transcription bubble. Then the RNA polymerase is going to join nucleotides to the RNA molecule at the 3' OH end. And finally, it's going to rewind DNA into the double helix form at the upstream edge of the bubble. So RNA polymerase does have some conditions that it works best in. The temperature really needs to be about 37 degrees Celsius for it to work. Any higher than that or any lower than that, the efficiency of RNA polymerase goes way down. And then, so this is going to be a pretty quick process, much quicker than RNA or DNA replication. So we can actually get about 40 nucleotides produced per second. This is slower than DNA synthesis. I was thinking transcription. So this is a process that does take quite a bit of time. And the reason that it takes quite a bit of time is because we're dealing with RNA and not necessarily DNA. So the third and final step is termination. So termination is where RNA polymerase is going to move along the template and transcribe the termination site. So that termination site, if you remember back to one of the first figures we looked at, it's going to be the red end of the coding region. And there's certain events that actually have to happen simultaneously to get transcription to end. So the RNA polymerase must stop synthesizing RNA, then it has to be released from RNA. The new RNA strand has to disassociate from the DNA, and then RNA polymerase must be detached from the DNA template. So those events happen very, very quickly, and a lot of times it's simultaneously. There's two types of terminators in our prokaryotic systems, and those are going to be the Rho-Independent and Rho-Dependent termination systems. Rho-Independent is where the gene does not need the RHO or Rho protein to actually terminate or carry out its function. And Rho dependent is where the protein is needed. And we're going to look at those more in depth. All right, so with termination, again, we've got two major types of termination, Rho-Independent and Rho-Dependent. Let's look at Rho-Independent first. So first of all, in Rho-Independent termination, this is going to contain, the DNA is going to contain several inverted repeats, repeat sequences, and these are going to be followed by approximately 6 adenine nucleotides, so 6 A's. Okay, so the Rho-Independent termination system is going to look for the inverted repeats, and those are going to be transcribed into messenger RNA. And then those repeats are then going to form this hairpin loop structure. And that hairpin loop structure is then going to signal the messenger RNA to disassociate from the DNA template. Okay, so if we follow this through the process, we've got our specific sequences followed by our poly A tail over here. And the inverted repeats are then going to be transcribed into messenger RNA. And those string of uracils, this is why uracil is such an important component of RNA, is going to cause RNA polymerase to come to, or is going to cause RNA polymerase to pause. So that's like an internal signal. And so RNA polymerase is going to pause, destabilizing the RNA and DNA pairing. Then the inverted repeats are going to fold into a hairpin loop, and the RNA transcript is then going to separate from the template, terminating transcription. So at the end of this process, transcription is going to terminate when the inverted repeats form the hairpin loop, followed by a string of uracils. All right, so that's Rho-Independent. Rho-independent termination is where the Rho, we do not need the Rho RHO protein. And so as we look at this more closely, we've got our adenine nucleotides back here at the end. Those are going to cause RNA polymerase to pause. We're going to get the formation of the hairpin loop, and then that is going to cause the mRNA strand to disassociate from the DNA strand. All right, so that's Rho-independent termination, and in the next segment, we'll look at Rho-dependent termination.